We were assigned case study 14, which is about a female patient who is 18 months old who has been in and out of the ICU. She received a variety of medications in her lifetime, and within the past couple of days, she has shown symptoms of a high fever accompanied by nausea and vomiting. Lab reports have shown an increase in white blood cell count. A CSF sample was collected, and from that we were able to isolate colonies of unknown 14. Our job was to figure out what the bacterium is, why it's causing these symptoms, and how to combat them. First, we examined the colony morphology itself, and we found that the bacteria were circular in form, with a raised elevation, undulate margins, and they appeared slightly off-white or beige. The colonies themselves were around 3 millimeters in diameter. We then performed a series of biochemical tests, and shown in Figure 2 are the results of the selective and differential tests. Note that in this image, the bacteria grew a colorless colony on the EMB plate, experienced complete clearing on the BAP plate, and had no growth on the MSA plate. Next, we performed 3 respiration tests, and from this, our oxidase test produced negative results as the bacteria did not react with the oxidase reagent. Our catalase test showed positive results as the bacteria experienced bubbling upon the addition of hydrogen peroxide, and our nitrate test showed that no gas was formed in the Durham tube. However, the addition of nitrates A and B resulted in a positive reaction as the tube changed color. Figure 3 shows the results of motility testing after 24 hours of inoculation. This plate was used to qualitatively determine if unknown 14 was motile or non-motile based on how much the bacterium spread from its stab point. Our enteral pluritest only showed positive results for glucose and urea, which are marked with orange arrows. Everything else was negative, and in the gas formations, we were unable to tell if the wax was lifted. Table 3 is a summary of the results of biochemical testing, as well as if each experiment was positive or negative for that particular reaction. Next are the results from gram staining, and note that the bacterium in this image are bacillus-shaped in single rods, and they turned purple upon the gram staining procedure. Antibiotic testing was utilized via the Kirby-Bauer method and MIC tests. On the left in Figure 6 is the results from the Kirby-Bauer testing, and on the right are the results of MIC testing. Table 4 summarizes the results of antibiotic testing. Note in the Kirby-Bauer testing that the ZOI increased as we moved clockwise, and the minimum inhibitory concentration was found to be 3,200 micrograms per milliliter. Finally, blast testing was utilized. On the left is an image of the 16S rRNA sequence of unknown 14, and in Table 5 on the right, note that the bacteria Bacillus cereus was found to have a 95% core recover with an e-value of 0.0 and a percent identity of just over 98%.
Before we analyzed our conclusions, we had to first determine the validity of our results. For each test, there was at least 2 other species of bacteria being tested alongside our unknown number 14. These bacteria were considered the controls and determined whether or not the tests were being performed correctly. For all of the tests, the controls were experimentally determined to be correct, and the outcome aligned with the information known about that particular bacteria. If our controls were considered valid, then it could be inferred that our unknown was properly tested too. This method is shown in Figure 9a. Once the tests were considered valid, then we researched the interpretation of the data, such as shown in Figure 9b, while looking at our lab manual charts. A source of error uncertainty that Emmy and I ran into was during our motility test. If you look at Figure 9c, it is unclear whether or not our unknown bacteria was motile. At first glance, it obviously is not as motile as P. mirabilis, but consequently also doesn't resemble the growth like S. epidermidis. We were unsure if our unknown was motile and impeded from spreading out due to its large colony size and waxy nature, or if it was non-motile and just naturally a much larger colony than S. epidermidis. From the evidence provided by colony morphology, biochemical testing, and BLAST software, we've concluded that our unknown bacteria number 14 must be Bacillus cereus. According to the BLAST test, our quarry coverage, or the percentage that our sample aligns with the reference sample, was 95%. We can also look at e-values for more evidence to support this claim. A low e-value is the best indicator that our results was not by chance, and our outcome for this BLAST test was a value of 0. Lastly, from the BLAST test, our percent identity was 98.38%. The higher the percent identity, the more likely the match is significant and not due to chance. There were 6 separate experiments performed for the biochemical testing. These tests included MSA, EMB, BAP, nitrate, oxidase, and catalase experiments. For MSA, or unknown bacteria species could not survive on the MSA plates, indicating that it wasn't a Staphylococcus species and that it couldn't survive in high concentrations of NaCl. For the EMB experiment, our bacteria also did not grow, which meant that it was a gram-positive organism. For the BAP reaction, our unknown lysis pattern matched beta-hemolysis by being completely clear around the colonies. We saw that with the nitrate test, our organisms utilized anaerobic respiration due to the fact that it turned pink. And lastly were the oxidase and catalase test. Our unknown bacteria did not turn black and purplish, indicating that it did not contain cytochrome oxidase, and bubbled in the presence of hydrogen peroxide, which meant catalase was present. Even though there was some discrepancy over our motility test, we came to the conclusion that our bacteria was not motile and that the colony was able to grow so largely due to the morphology and characteristics of the given unknown. Our enteropylority test came back negative except for glucose and urea. The positive reaction for glucose means that the bacteria ferments glucose to either acid or acid and gas by turning it from red to yellow. Our urea column turned pink from the original beige, which indicates that the bacteria utilizes urease to hydrolyze urea to ammonia. When looking at our Kirby-Bauer test, both erythromycin and amoxicillin were considered intermediates, which means that the resistance could be based on the dosage of the drug, where our last two were susceptible to the antibiotic, which means that it couldn't grow if the drug is present at all. Our minimum inhibitory concentration was 3,200 micrograms per milliliter, therefore the lowest concentration of the antibiotic that prevented visible growth of the Bacillus cereus. When comparing the unknown number 14 to Bacillus cereus species, the two bacteria match on almost every column except for motility, citrate, antibiotic resistance, and minimum concentration. We already talked about our discrepancy for the motility test, and we think that our sources of error for the citrate, MIC, and antibiotic testing was purely due to uncertainty in some of the internet images. Relating this back to our case study, there are two common types of illness from the Bacillus cereus, either a diarrhea strand or an emetic strand. Based off the symptoms given from our case study, we believe that this aligns with our predictions of the emetic Bacillus cereus due to the vomiting and nausea mentioned by our patient. Common treatment for this bacteria would be the use of the antibiotic vancomycin to kill the infection.